every day in a daily basis, the data generation rate in Edinburgh Institute is 800 terabytes every week. So I don't know how they do it, but it's crazy. Like the data size is crazy if you do this image with optical microscopy. And the other thing is, this is actually the courtesy that the mouse brain is provided by Professor Gosonghan here. So you will see that the same mouse brain, the same type of mouse brain, can show various immunostaining results after the same protocol. But the problem is the data we want from this image is the number of these single cell points. So you really need a good way to identify and to quantify how many of these small dots in every brain, and do it automatically, because there are thousands, tens of thousands of cells there. You don't want to do it manually. So to do that, you have to go through a lot of pre-processing methods and then going through this deep learning method. Currently, our lab is using NUNet, and it's really powerful. And even you don't know how to build this deep learning method, I will also suggest that you guys go to use. I think everyone uses ImageJ here, right? So there are several tools that you can use ImageJ easily. They have the very beautiful UI that helps you to do a little bit deep learning analysis. Their network is not so deep, so maybe not too complicated work, but they still help a lot. So by the power of NUNet gave us, we're able to segment a lot of these complex signals out of the entire image. And next thing is we want to extract the features. Like, for example, this is blood vessel. So after the segmentation, we wanted to see like how many branch points there, and what's the length of it, and what's the radius of each blood vessel, because sometimes for the some type of disease, the radius might change a lot. Okay, and the last part, particularly for the brain, we wanted to registration. We still wanted to know which part of the brain is changed. And I just wanted to see the difference before and after we have the deep learning model. So before we have a deep learning model, there are some ways to do analysis. Like you can choose a little block in the brain and to do this 3D visualization with commercial software. It's easy, but if you want to apply to a whole brain, it's impossible. And the problem with this process is totally, to do this, we kind of gave up all other information we get from this data. And it's super stupid, right? Like, even though you can get really beautiful, statistics here, but still, like, you are kind of choosing the region of interest, the volume of interest yourself. And of course, you were able to see the detail morphology out of this, like this. But still, you're kind of choosing, like by yourself, choosing the specific area by yourself. It's not subjective at all. So after you get these, sorry, this is done by my undergrad talented student. Like they want to make a movie out of it. Okay, so after you have this whole brain vascular segmentation tool, you can do whole brain and you can choose specific brain area. Like this is a hippocampus. And then you can analyze the entire vascular system in the hippocampus. So now it's brain region specific. It's not, human intervent, like human selection basis. Okay. Okay. So in addition to neurons, we are able to label the C-force activity by immunolabeling. So to do that, or we can just either genetic induce the C-force activity and make them become purple, or we can like label the protein directly. And after doing that, this is also the data provided from Professor Guo Conghan. So we are able to quantify from different group of animal and analyze what's the difference between these two group of animals. And most importantly, now we can do this whole brain system-wise analysis. But I just present this work 5 days back in Korea in the neuroscience meeting, a computational neuroscience meeting. I just told these guys that I have this, but I don't know what to do next. So if you are interested to do what to do next, you must to work with talented theoretical neuroscientists. They are amazing people. They're using math to, solve whatever math that I created here. Okay. So this is just another example that if we wanted to analyze dopaminergic neuron before we have AI, we can do some morphological analysis. But after having this code from this model in NUNet, we're able to quantify each cell. This is the movie that you guys saw already. So most importantly, you can now do colocalization. Like you can identify if the dopaminergic neuron is activated or not activated after your treatment. So this is really fresh data. Please don't take a picture of this one. And so this is the data we just generated like 2 days ago. You'll see that after some treatment, the C-force activity doesn't really change across different groups, and the dopamine neuron number doesn't change. But the activated dopamine neuron number changes, and in different trend, in different area. So you can imagine now if you're quantifying entire brain, it won't give you any data, right? You have to look at different brain area and see how different they behave. Okay. So I'll just quickly go through all the applications. So in 2021, we collaborated with Dr. Lin Jinxian and work on their Parkinson's disease mice, and then use these whole brain analysis to do dopamine neuron quantification. And 2 other papers, we work with Professor Hu Shangshou in our department. He's really genius on making different materials to treat different kind of disease, like from brain tumor to brain, traumatic brain injury. So what we did in the paper is that we also use this whole brain imaging tool to visualize how these neurons or how these neurons or how the brain tumor is being treated. And one most recent collaboration is between our lab and Professor Song Xingwen in Huagongxi. And so what they're interested in is to use oral injection instead of how do you say that regular injection. So they wanted to develop drug that you can take daily orally instead of taking shots every day. So they want to know if their drug is being taken and if the mRNA is highly expressed, being absorbed by the intestine cells. And indeed, we help them to visualize that, because with thin section, it's really, really hard to find these green dots. Like you can imagine if I have only one single slide, you won't be able to tell it's a noise or it's real data. But with the volumetric data, you're able to see that. Okay. So one thing you can do with AI is enhance resolution. So remember, I said that we can do super resolution with optical microscopy, right? Not only in lateral resolution. In axial resolution, it's really poor compared to the lateral one. So Professor Zhang Han in NTU actually developed this game model, the generative AI model, that can help us to do deconvolution in a very neat way that you can really see how much better the resolution enhanced in axial axis. Okay, so there's still a lot of things have to do. For example, we are still struggling on registration part. So after the whole process of making the brain transparent, making the brain multiplex, their shape has to change somehow. And every brain is different as well. So to find a really good way and accurately, register the model to every brain is really tricky in this process, and nobody can do a really good job now. And it's really painful to train different model for different target proteins. Like we do dopamine neuron, we have to train the model to segment dopamine neuron. If we wanted to quantify the extracyte, we have to train the model for the extracyte. If we wanted to do CFOS, we have to train the model for CFOS. It's crazy. Like nobody have this endless time to visualize and to quantify, to develop, to train so many different models. So we really need some universal model that allow us to see things together at the same time. And it makes more sense because, for example, when people do this image segmentation in the car, you see different things pop up instantly, right? You don't feel there's a delay between different object detection. So we wanted to detect different objects in the brain simultaneously. And analyze part. So I don't have a good way to analyze these data yet. We're really eager to learn from the MRI people that they have already built up a system to do the brain image analysis in a whole organ-based perspective, but they're using different data as us. So I still think there are much more to read, to understand with the data we acquired. And of course, for the live imaging part, which I didn't mention today, we still cannot do whole brain activity image in the cellular resolution, so this is something still needed. And finally, if there, this is the slides I used last week in Korea. So Dr. Pike is one of the prestigious researchers in theoretical neuroscience, and he believed that if we have enough information from structures, we might be able to understand the brain without understand each neural activity at the same time. And that is so important because after you learn how our brain works, we want to build something, and build something by means that we only know what is the hardware looks like, but we don't know how the information will looks like after. That will only happen after we build it, right? So, for example, like if you wanted to understand how to build a city, you will analyze the current city that have people living in, right? But then you get the idea that how the road and how the design can affect people's behavior, and then you build another city, and then you hope that is best for human living. So I think that'll be it. And we also have a startup company that's going on for 5 years. They are helping people to do this 3D image as well. And I want to thank all the talented students in my lab, and of course the brain research center that led by Professor Zhang is giving us great support to do a lot of these things, and all the great partners like Professor Guo and Professor Lo and Professor Sun in the past few years that helping our team to really grow and can do some cool stuff. Okay, I think that'll be all. Thank you so much.